righty. Looks like people are starting to pour in. Okay. So in the interest of time, I'll go ahead and introduce you, Steve, and then as people pour in, I'm sure we'll see more signs. Welcome everyone to the Health Application Society online seminar series. We're really excited for our speaker today. Dr. Steve Chick is a professor of technology and operations management at INSEAD. He was named the academic director of INSEAD's Healthcare Management Initiative and the Novartis Chair of Healthcare Management in 2008. He received his MS and PhD from UC Berkeley in the IEOR department and BS in math from Stanford. And before his current position, he was a faculty member at the University of Michigan's IOE department, Go Blue. And he had worked for 5 years in the automotive and software industries. So his work brings together operations management, simulation, statistical decision-making tools, among others, to help improve healthcare operations, as well as the assessment of new health technologies. And he's been very involved in Informs. He's held all elected officer positions within the Informs Simulation Society. He's been active in many editorial roles and is a past president of Haas. So we're excited. And then he's also an Informs Fellow, which is a huge deal. So we're super, super excited to have you, Dr. Steve Chick. And with no further ado, I'll let you take it away.
Great. Thanks so much, John. And thank you, Malika. I'm really happy to be here. It's an honor to be part of this seminar series. And I'm really looking forward to spending time with you. And thanks for the mention of Michigan, Go Blue. Actually, my mom was a student there when she was pregnant with me. So my time from Ann Arbor goes way, 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 way back. But now I'm Go Green. I'm at INSEAD, and green is our color here. And it's a pleasure to share with you some of the things we've been doing through time. Actually, thinking about what to talk about was an interesting question. I gave a talk maybe 5 years ago at the Winter Simulation Conference, and that was a plenary, and that's recorded. That's on the WinterSim website for 2020. And that was in the area of looking at which type of mathematical model to use for resource allocation decisions in healthcare. Done a lot of work in epidemic modeling, discrete event simulations, stochastic models. And we were looking at, well, how are the decisions made on the basis of those models dependent on which model is chosen? And does it matter for any given application, yes or no? It turns out that the type of model you use might change the decision you make. And that's something I did a lot back in Ann Arbor with Jim Coatman. We called it model transition sensitivity and analysis. And that was really motivated by Jim's amazing work in the science of disease transmission and control. And I'd like to thank him for his friendship and his scholarship and all he did in my life. He passed away this past December. So thank you, Jim, for all you've done in my life and for what you've done for the world in public health. I'd also, another topic I was thinking about talking about was in the area of some of the interventions we've done around leadership and innovation and executive development for people working in healthcare systems around the world. That's kind of a little bit more on a teaching side of things. And I was really lucky and fortunate to be able to do that thanks to the support of the Johnson & Johnson Corporate Citizenship Trust, and in particular through a relationship developed with Frank Velvert. And Frank was very instrumental in the development of my teaching here at INSEAD and health impact in practice. Frank passed away just this last month. And I'm very thankful for everything he did in my life. So thank you, Jim. Thank you, Frank. You've made a difference for a lot of people around the world. Today, I'm going to talk about neither of those topics. I'm going to talk about some work that I've been doing over the last decade around clinical trial design. How can we rethink even how we approach clinical trial design rather than a cost center, think about it as a value option to create value? And that's been some work I've done with a lot of colleagues at a lot of universities and with some funding from a number of different sources. So I'd like to really thank them all. The seminar is only an hour, so I'm not going to read everybody's name. You know who you are, and I'm very grateful for all we've done together. Standard disclaimers here as we go forward. The views that I'm going to express are mine and not necessarily those of the National Health Service of the UK, National Institute of Health Research, European Union, and other supporters and collaborators in our organizations. And I will be giving some graphs that show a few things about healthcare. These are numerical examples to illustrate ideas, and they're not to be constituted as medical advice. So with those thanks and with those caveats out of the way, let's dive into today's webinar. I'm very happy to be able to speak with you about this really interesting issue. I think a lot of us have understood that clinical trial processes can be very, very expensive for pharmaceutical companies, and a lot of us are concerned about the cost of clinical trials. This is a decade-old data, 1.7 billion US dollars per new molecular entity on the market. And you can see there's a lot of preclinical phases, a lot of clinical phases, phase 1, phase 2, 3 trials that go into that, and they all add up. And you got to do a lot of early stage exploration to get that one hit. But I mean, this is very expensive. So a lot of people are worried about how do we address the cost of that. Now, one of the observations I had in starting to look at this was observing that phase 3 trials are really about identifying effectiveness, medical effectiveness relative to some standard of care. But often in many countries like England, you want to look at incremental costs and incremental benefits. And is that in a good balance? Are we getting good health value for money? And as we all know in operations community, if we have a flow of goods and we have different places along that flow, different operations along that flow, and they have different figures of merit, there's an opportunity for misalignment. We see that in double marginalization and supply chain. Is there a similar issue going on in clinical trials? And can we do something about that? Is there a suboptimal flow of ideas to market in terms of health value for money? And so that's what motivated me to start to look at that. Now, the example I've given here is it's about drug development. But there's other health technologies, and I'd like to speak about health technologies more broadly, not just drugs. Think about devices, think about changes in care processes. Maybe there's not a new patented technology, you're just changing the way you're delivering care. Some of these are going to be privately funded, like drug development typically is, but there may be publicly funded trials just to see how triage works differently in hospitals in the UK, for example. So a lot of different types of trials. And a lot of people care about this, right? People who run trials care, regulators care, payers care. NHS England 8 years ago uh, actually highlighted in some of their documents, it's important to bake in the value and real world costs of research as a default part of funding projects. A couple of years later, the UK's National Institute for Health Research, the equivalent of NIH for the UK, was looking to stimulate research to try to improve the design, development, and delivery of more efficient, faster, innovative studies to provide robust evidence to inform the clinical practice and policy. And it's those trials that then inform technology adoption decisions. Do we want to buy that stuff at a national level in the national health system? How much should we pay for that? And it's not just UK. Other countries in Europe, the European Medicines Agency is interested. US FDA has given guidance about how to do more creative clinical trials through time to try to address some of these challenges around these big costs. Clearly, the for-profit pharmaceutical firms, venture capitalists that invest in biotech, they're also very interested in addressing this problem. Okay. So if it's such a big problem, has anybody looked at it? Well, of course people have looked at this. There's a lot of people who've thought long and hard about this and come up with really cool solutions. A lot of them are focused on reducing the costs of clinical trials. There have been adaptive trials. There are multi-arm trials where you're testing several different alternatives all at once against one placebo, so you don't have to repeat placebo groups. Group sequential trials, let's collect data. Rather than collecting data on 1,000 people all at once, let's collect 200, 200, another group of 200, and then check the data, see what's going on, and see if we can cut back the sample size if the signal is very positive or very negative against this new against a new treatment. And typically the approach there has been you minimize the expected sample size of the trial, subject to some statistical guarantees, type 1 error, type 2 error. You got to get your statistical power. And so that's kind of one of the things that a lot of people have been focusing their attention on, and very successfully. Um, Delay in outcomes is a big thing that we often see in clinical trials, and there's a very neat paper by Hampson and Jennison in JRSSA that was talking about, well, how do we minimize sample size, given that we know there's delays in observations? We treat the patient now, we see the outcome 12 weeks later. How do we make sure we get our type 1, type 2 error? Very nice stuff. One of the neat things I think in some of the statistics journals is you get commentary. We don't always have that in management science or operations research, but you get commentary on some of the articles. One of the commentators, Berman, was saying, can't we use Bayesian techniques instead of this frequentist approach to incorporate the information about the patients, the pipeline patients, the patients we've started to treat, but we haven't yet seen their outcomes because that 12 weeks time hasn't elapsed till we see the outcome. David Draper was suggesting, why focus on 0-1 outcomes, treated, successfully treated or not? Why not think about quality adjusted life years? Let's think about the health gain associated with a new treatment. And so, there's a lot of interesting dialogue there. Well, the bringing in of this Bayesian and quality adjusted life year is something that some people have done for follow-up studies around health technology assessment. So when the phase 3 clinical trial is done, somebody's going to look at the costs and the effectiveness and try to figure out, well, how much additional information do we need using expected value of sample information ideas to see how much additional information we should collect in that kind of evaluation phase after the clinical trials are done. But nobody was really kind of pulling that type of idea back into how do I design the clinical trial itself. Laura Flight in 2019 with several co-authors doing some studies of adaptive trials and thinking about this value-based value of information approach was noting that a lot of people were excited about that idea, but not a lot had been done to that date. And so we were trying to work in that space. So rather than think about it as a cost driver, can we start to think about a clinical trial as an option to learn to see whether we want to adopt a new good treatment or whether we should stick with our existing treatment? So let's think about value, and we'll balance the cost of the trial against the expected value of the information that's coming up. Now, we're dealing with health and we're dealing with money. And so we need to have kind of common unit, or that's one approach would be to have a common unit. And one way to do that that's kind of commonly used is to say, well, let's take a quality adjusted life year gain, multiply it by some fudge factor, say 30,000 pounds per quality adjusted life year, and have that represent money. If we're in that space, well, how much data should we collect? How can we adapt the number of patients we see? How do I adapt my sample size? And that's going to where we'll start. And we'll talk about how to randomize if we have multiple arm trials. We'll talk about precision medicine trials a little bit later to try to customize treatments to patients as well. But that was kind of the motivator for the sequence of work that I've been driving and just having fun collaborating with others with. And that is, can we better align clinical trial design decisions with clinical trial designs with the decisions that are going to be made as a function of the trial data? So that's where we are. Now, healthcare is a complex place. You all know that. So as a modeler, I tend to go about things with modeling rather than empirical to start with. And so I like to have a simple model to start with. So I said, well, let's just start with, assume we can sequentially observe data. And we just want to figure out, well, when do we stop collecting data and then make our technology adoption decision? Yes or no? Is this a good treatment, not a good treatment? And well, we tried to do that, bringing in these concerns of Draper, which was accounting for the pipeline, the delays and the observations, and Berman using the qualities for quality adjusted life years. And it tried to incorporate a lot of the work that we had done previously around Bayesian sequential optimization in the discrete event simulation community. and building on some of the cool stuff that Herman Chernoff had done way back in the day around hypothesis testing on Brownian motion. So we'll bring in some of that. Right? So that's kind of the mathematical framing and kind of where we're going to start. Two-arm trials, compare a new treatment with an existing standard, and we're just going to figure out how much data we should collect if we can observe some data as we go along. In deciding to make that early stopping decision, we're trying to balance saving resources. We're going to save costs from the clinical trial, and we can use the savings to treat other patients or to further research in other areas. On the other hand, the countervailing balance is if we collect too little data, we might make a bad decision because we don't have enough information to know what's the actual best treatment, this new standard, the new or the standard. So for better or for worse, that's where we started. And what we ended up with was a paper that looked at fully sequential sampling, not group testing 200 by 200, but you look at two patients at a time, one to the new, one to the standard, look at the differences, and then just see how many patient pairs you want to enroll. And you can observe data as you go along, but there's a delay in observing the data. So this is a Bayesian stoppable bandit with delayed observations. And we're considering this as a single project where the expected health benefit is converted from quality adjusted life year gains of the new treatment, potentially, minus the cost of that and minus the cost of research. And I'll explain how we get there because I know that in Inform's crowd we want to see how does it work and what's under the hood, at least at a high level. But I give you a little bit of a punchline. My kids tell me I'm a bad joke teller, and I'm going to be a bad joke teller here. I'm going to tell you the punchline right away, and then I'll explain the joke after. Minimizing expected sample size, which has been a driver of a lot of those things we've been looking at, including the National Institute for Health Research call for innovative trials to reduce the time to get drugs to market. That's not the same thing as maximizing the expected health benefits. We're optimizers. We know that if we change the objective function, we might not have the same outcome. So maybe that's not a surprise. But I think it's a shift in thinking about that. Efficiency should be relative to the amount of money spent to improve exploration to find new health treatments, rather than in terms of the cost of just the trial itself. So I'll share with you kind of where we kind of landed on that initial model, and then I'll take you through 3 Re's: relate, reinforce, and reward. So we'll develop the base model for 2-arm trials with optimal stopping. It's going to be a little bit theoretical. And then, well, theory's fine, but will it work in practice? So I'll share with you some observations about some attempts that we've made to see how this would work in the UK National Health System, in the National Health Service. We'll try just for the 2-arm trials. We'll then show what we've kind of at a high level, what we've done to kind of look at a broader set of trials, multi-arm trials, precision medicine trials, where a drug might work for certain people with a certain biomarker, but not for all people. And all of this is going to be based on a social perspective. We're trying to maximize health gain minus costs. We're not looking at for-profit incentives of firms in that. But the reality is that companies want to make money as well for developing and delivering new drugs. So we need to think about those incentives. So we'll think about rewards, market access, conditional approval schemes, and pricing negotiations. How would you manage a portfolio of that if we have time? Okay, so that's kind of the outline of what we're going to think here. So first steps. I'm going to try to highlight kind of the clinical trial space is too complicated, right? So many different types of trials. And I'm just focusing on like phase 3 or phase 2, phase 3 trials and the technology adoption decision to keep it simple. I'm going to put in blue the assumptions for the base simple model. So I'm going to look at a single trial to start with, rather than a portfolio of investments in a multiple set of biomedical innovations. I'm going to assume that early stopping of the trial implies I can make a decision faster and it will lower the cost of the trial. I'm going to assume that I'm going for a social planner's objective, UK National Health Service, working with the Department of Finance, looking at investments in care and in new drugs, and accruing the health benefits, endogenizing those benefits to the tune of 30,000 pounds per quality adjusted life year, for example. Because I'm looking at quality adjusted life years, I need to have real valued outcomes, not 0-1 outcomes. And I'm not going to look at trials that are monitoring trajectories through time per se. I'm going to assume that outcomes are normally distributed, as many of us all often do, right? Normally distributed in the same units as sampling costs. So I'm going to assume that there's two arms. I'm going to randomize at each time step two patients to the new and the standard. I'm going to look at their outcomes. I'm going to subtract the difference in their outcomes, and that's going to be my basic observation. I'm only going to adapt the length of the trial and not going to adapt the fraction of patients to each arm. I'm going to assume that the delays of interest are such that I'm able to respond in a reasonable amount of time. So almost immediate observation, weeks or months, maybe even a year. But I'm not going to really address issues like 5-year oncology survival studies. That's beyond the scope of today. I'm going to assume that we're really interested in getting as much information as possible to make a decision and have a bunch of post-trial patients, say the next 10 years of patients who might benefit from this procedure. So it's an offline learning, offline rewards, if you will. And in this base model, I should mention, is with my good colleagues, Martin Forster, who is at University of York. He's now in Bologna, and Paolo Pertile in Verona. So it's a lot of fun working with them on this project. I think I mentioned that kind of our basic observation, we're going to make randomized to the new treatment to one patient, the standard of care to another patient. Look at the difference in effectiveness, multiply that by that factor. But I just wanted to put a little notation there so that we can grasp that. We also need to look at the treatment costs. And it's not only the cost of the drug, but the cost of complications, cost of the whole treatment process. And we want to look at that difference. And this thing is called the incremental net monetary benefit in health economics. And so we're going to assume that's our basic observation. We're going to assume that we don't know the mean. If that mean is positive, then the new treatment is better. If the treatment is negative, if the mean is negative, then the standard of care is the better alternative. And we'll do our usual Bayesian thing, put a prior distribution on the unknown mean, and we'll work on that basis. After all of the data is in, after all the trial data is in, we have a second decision to make. So in addition to deciding whether to continue a trial or not after each observation, we're also going to decide at the end of the trial, do we adopt this new technology? Now, we're going to adopt that new technology if, say, for our post-trial population, say 100,000 people, big P is 100,000 people to be treated that are going to be affected by this decision of, do I treat with this new technology or not? If the net health economic benefit in expectation is greater than the cost of implementing that new technology in practice. So if we want to go to this new, we have to deploy this to hospitals, we set up procurement agreements, train people. So there might be a fixed cost there. As long as it's looking good, if the new treatment can cover the cost of changing, let's do it. So that's the basic idea here. Now, how do I see this trial actually running? So we're going to start the trial at time 0. And let's suppose we're in that setting where it takes 12 weeks to observe the outcome. Right? We start a treatment process, and after 12 weeks, we know, is a patient doing well or not, and how well they're doing. And suppose we can recruit 10 patients per week to enroll in the trial. That means at any given, we're going to have to enroll 120 patient pairs, 12 weeks times 10 patients per week, in order to start seeing any data. So there's going to be a period of time where we get out to say tau is 120, where we're not seeing any outcomes, but we're enrolling patients. Then there's going to be a next phase of the trial where we're observing data from the initial patients, and we're still choosing to randomize more patient pairs into the trial until we get to a stopping time, big T. And after that point in time, we don't enroll any more patients. We just keep observing the rest of the data that's out there, and then we're going to make a decision, new or standard treatment, to adopt at a national level. That's at time big T plus tau. Okay? That's kind of the flow of the process. So three main phases: randomizing without seeing data, randomizing with seeing data, and then kind of waiting for all the data to come in. Okay, so that was the cute picture. Always very helpful for me when I'm reading a paper is the cute picture. Then comes the math, and I know we all love the math too. Let's just put a bunch of Greek letters. The main Greek letter of interest here is the following. The posterior mean, given that we've randomized little t patients so far, that's going to be a very important random variable for us. And then our policy pi, that's how, our policy is do we choose to continue or stop the trial? And then once all the data comes in, um, which technology do we adopt, the new or the standard? And we can allow for discounted rewards or not. And basically, the bottom line is we want to try to look at, well, what's the reward of any given policy, the research costs, which is a random variable. We have an adaptive stopping time here, and the rewards from adopting. If we stick with the standard, declare that the standard is something we should stick with, we get 0 for the adoption rewards. Otherwise, we get some positive amount. The better the treatment, the more reward we get. So we're interested in finding the optimal policy to maximize that reward. Because of the structure, Bellman's equation comes to play. We can do a bunch of nice structural results, prove various optimality results. But I won't go through those. The paper's got a big pile of them. We need to compute stuff, right? So I'm a big fan. When I was doing the department editor of simulation at OR, I was really encouraging people to make their code publicly available. And I know that that's something that management science is really doing a lot now. I'm really encouraging everybody to, expose your code. Here's my code for this paper and all the other papers that I'll mention a little bit later. It's available for you to play with. I think it helps our community grow faster and helps us learn from each other. Okay. So what does a policy region look like? Just to kind of get a feel here. Okay, so here's a graph of the optimal stopping boundaries. On the vertical axis, I've got the posterior mean based on all the data we've seen so far. Typically, when we start a trial, we're going to assume that prior mean is 0 because we're not sure if the new treatment is better or worse. Our effective number of samples in the prior distribution is down here. And then as we add observations, so we're going to go along the horizontal axis. In this particular case, let's suppose we start out at 0 with our prior distribution. That's right here. We start randomizing data. We're randomized about 1,000 patient pairs, and then we start seeing data because our tau is about 1,000. And on the black line, our posterior mean goes up and down until we hit this dashed line. Our dashed line is our optimal stopping boundary. As long as we're within this region here, this yellow region, we're going to keep continuing to sample, continuing the trial. As soon as we get out, we're going to stop the trial. We're going to wait for the red sample path to finish, and we're going to then see, are we positive? Are we doing good? Or are we doing bad? High is good for the new, low is good for the existing standard on that axis. So this is kind of the flavor of that. What's more, so that's operational and interesting for somebody running the trial. For somebody planning the trial, there's something else that's even more interesting, and that is with this you can compute monetary values. So suppose our prior mean is 0, given that we have no data so far. If we want to look at what is the expected value of a trial that's run fully sequentially versus an existing trial, we can compute that using this Bayesian approach, value of information. We took some data from a comparison of drug-eluting stents versus bare metal stents adopting in the US, assuming on the order of 2 million adopters of drug-eluting stents or bare metal stents, should that be the better alternative. And what we found was that you can compute the value of this response adaptivity going up to our jagged line here is on the order of 2 times 10 to the 7th dollars. That's $20 million in expectation if you can endogenize the health gains from a better treatment with drug-eluting stents. Now, for 2 million adopters, that's $10 per person. At the time that those studies had been done previously, the medical studies and a technology adoption decision, it was about $1,500 per stent. So that means you can save 0.2% on the cost of goods sold in this particular example. Now, any manufacturing plant manager would love, in any industry, would love to save 0.2% of their cost of goods sold. And this is from just running a response adaptive trial, assuming you cannot endogenize the health value for making a better decision. So that's the theory side. It can prove a bunch of structural results there. But that's how does this relate in practice? Now, our colleague Martin Forster on this paper was at the University of York at that time, and he had some friends at the University of York Clinical Trials Unit. So we started talking with them to see, well, how could could this get airtime in the real world here? And so the clinical trials group at York was kind enough to share with us something that they had done about the Proffer trial and health technology assessment, a trial that they had done. And they said, well, why don't you kind of do a retrospective analysis of the data there and try to see how would it play out? Could it be done? And so they had this data is about proximal humerus fracture. So this is a particular type of bone break, and that can be treated through surgery with a plate and some screws, or it can be treated with a sling with immobilization. So how would it play out? So we took a look at the data that was available at the time at the start of the trial. We looked at the actual budget for the trial, which is on the order of 1.5 million pounds. We looked at the data that they proposed to collect, which is about the Oxford Shoulder Score. It's a measure of mobility of the arm, which we can then convert to QALYs and then convert to money. And let's see what happens. So the citations to a couple of papers are at the bottom of the page here for you if you'd like to follow more details. So let's see, what does this look like? Well, what we had was the actual cost data. How was money spent through that trial? So recruitment for patients of the trial started in late 2008. And data was, the costs accumulated through time throughout the trial up until the 1.5 million pound budget. At time B here, so just in the spring of 2011, recruitment finished. You could see outcomes of the data, the 1-year follow-up in early 2012 from the last patient. And then you can see there was some additional follow-up to 2 years and then some publication going on. So this is the cost profile. Okay, well, that's interesting. Under the traditional clinical trial approach, you'd wait until early 2012 before making the decision, is this new treatment, is sling better or surgery better? At that time, 1 million of the 1.5 million pounds had been spent. But there had been signal accumulating since early 2010. Was there a time where you could decide to stop the trial early because it's not worth collecting that extra data? So here's this is the first graph that we were aware of, where you're plotting costs with the axis on the left, the label on the left, and on the label on the right, what's the cumulative estimate of the incremental net monetary benefit given the patient data that had been observed? We can start to look at that. So we took the data that was available at the start of the trial and tried to figure out, well, what would the optimal stopping boundary have looked like? And that's this blue line, if you were to allow the trial to continue to twice the length of the trial that had been actually run. Actual trial had 125 patient pairs. We said, well, maybe you should take more. So we allowed the maximum to be 250. The black line here, the black solid line that I'm now highlighting, that's the actual sample path of the actual trial. And we see that sample path stopped well before the 125 patient pairs were actually needed. That's going down, which means that that trial was recommending that sling was more cost effective on the basis of that data. Again, I'm not making medical recommendations. I'm reporting these to illustrate the concept. So we took the actual patients and we kind of generated a bootstrap of all the actual data process of the actual trial. We found that on average, 75 patient pairs were needed rather than 125. You could save 15% of the cost on average of that trial by using this kind of approach, this value adaptive approach. And it turns out that 92.5% of the bootstrap trials agreed that surgery was not cost effective relative to sling. So sometimes it got a different conclusion, which meant that maybe it was losing some value. But that's we showed that you could actually do this. This is something that's feasible to do, at least in one trial. Now, how can it work more broadly? You don't want to overstate your what you've achieved. We tried to play with a few more types of trials here, and we played with um, well, we got some funding. Laura Flight, who I mentioned earlier, who had done some work around can value-based thinking apply into clinical trials design. She was the postdoc on this project with Alan Brennan's group at Sheffield and the York Trials Unit, and we found some interesting stuff along the way. We found that with the Proffer trial, yeah, you can shorten the trial a bit, but the actual net monetary health gain from making those decisions is not hugely bigger. It might not be worth it to run as a value-based trial there. We tried another trial, which was around hydroxychloroquine for osteoarthritis. There we had to figure out, well, how do you deal with missing data? And so that was actually Martin Forster and Charlie Welch and a few others at the York Trials Unit. They found that you have to deal with imputation of data. Somewhat longer trial was better, but again, not very much health gain in terms of health value gain. So that was a bit disappointing. But you don't know. So with Laura and with Alan Brennan and a few others at the York Trials Unit, we looked at the cactus trial. And there it was very interesting because we found that not all trials have a nice, neat observation you can have at the end of the day. You have to process it through a health economic model, and you use that output of that rather than the output direct from the patient, as I made it simplified it earlier. But in this case, it was very interesting because the optimal would be to run a much longer trial than was actually run in that real scenario. And there could be a big potential gain in value, health economic value. If you think about the improvement in health, because you've got more information, you make a better decision, there could be a big victory there. And so for one of these three trials that we studied in depth, we found that this value adaptive approach could be very valuable relative to assuming the goal is type 1 error, type 2 error, and minimize the expected sample size. So I would really ask us to reflect, is efficiency in clinical trial relative to sample size, as many have proposed, and could be a very useful tool. On the other hand, maybe efficiency means efficient in getting new technologies to market. And it might mean we have to take a longer sample size. That's a couple of observations there. I think there were two other things I think that are worth mentioning here. One is about trust. One of the trialists at York asked me, well, how do I know your code works? Why should I believe your code? You've done this MATLAB code. It's not in my standard packages. And fortunately, my colleague Paolo Pertila had actually done an independent implementation of the mathematical model, and we were able to show that, yeah, we're getting both the same output, the same recommendations, and so that was a reassurance. So building trust and exposure to code was a useful thing in that process. Another thing we found was an organizational thing, and that is if you hire somebody to run a clinical trial for, say, 3 years, and the contract runs for 3 years, and your funding is for 3 years, if the response adaptive trial says you should keep going with the trial, well, maybe some people are going to move positions that causes disruption. So it might be that response adaptive trials, when they go long, is less implementable than if they go short. If they go short, you can have somebody work on a different project. But that got us thinking, maybe if we don't just response adapt the duration, but we response adapt how we're allocating to different arms. Maybe that could be something that's more easily to implement. So make sure that the value of information for each randomization decision is helpful. Maybe that would be a good thing to do. So that's what we looked at next. We tried to reinforce, push beyond the two-arm trials into a broader set of trials, like dose-finding trials, or highly multi-arm trials, where you're mixing and matching different combinations of treatments as part of a treatment bundle. In that case, you don't want to have to randomize one patient to every arm. There might be a combinatorially large number of arms. Or in dose finding, you don't want to randomize to people to high doses first. You want to randomize them to low doses first in case there's safety issues. So you're going to lose that notion of having one person on every arm. So you're randomizing to individual arms at that point. But we found that the approach, the expected value of information per unit observation, that can work. You have to worry about finding a good prior distribution for the unknown rewards of the different arms. Some of those rewards are correlated. So we've given some guidance regarding how to develop a pilot study, manipulate data from the pilot study with empirical based methods and some tricks that Ilya Ryzhov and Warren Powell have documented in their book are on optimal sequential learning, really neat tools. We had to adapt that here. And it turns out that it works quite well. So in this graph, low is good, and our algorithms that we were able to find are lowest on the curve. Wrong, on the vertical axis, it's the expected opportunity cost on a log scale. On the y-axis is the sample size. So seems to work well. I don't want to go too much into that because I think there's another challenge in trials that I think is much more potentially has a potential for much greater impact, and that's precision medicine trials. A lot of the biomedical innovations that are going on today are around trying to find biomarkers and finding treatments that go to those specific biomarkers. This has been very, very successful in oncology, for example. And we had the good pleasure of working with some of the folks at Academic Medical Center, now University of Medical Center in Amsterdam. One of my former MBA students is heading up the ICU there. Shout out to Alexander. And so we started looking at some issues around sepsis diagnosis and treatment. Sepsis, I hope none of you have had that experience of having a loved one or yourself with sepsis. Very strong disease, big killer, number one killer in hospitals in many developed countries, and a very high cost financially. So the team there in Amsterdam had collected a bunch of data on hundreds of patients regarding when they were diagnosed with sepsis, their outcomes, a bunch of data about their blood. So blood transcriptomics, Brendan Skikluna took an analysis of hundreds of observations of different biomarkers in blood and found they clustered into 4 areas called them Mars 1, 2, 3, 4. Apache score, they had data around certain features of disease severity. And they were interested in exploring 8 different types of treatments: high and low fluid, aggressive or restrictive fluids, tight or loose glucose control. So 8 different treatments here, mixing and matching. Interestingly, what we found with Andres Alban, who did his PhD here and is now in Frankfurt, we were finding with the Amsterdam team that people with Mars 3 tended to respond differently to aggressive fluids and non-aggressive fluids. So that would be when there's an interaction between a treatment and a particular biomarker, that's called a potentially predictive covariate. And there's other covariates like Apache score. The higher the score, the worse the outcome, no matter what. And so that's a prognostic score. And so in many cases, you have some sort of a knowledge or some hint that there might be a particular structure with respect to what is predictive and what's prognostic. So our question was, can we use that knowledge to do a better job of being efficient with our trials? So we'll have a fixed length trial, but can we use those resources more efficiently to try to rule out which treatment is not good for a particular patient and which treatments would be good for other patients? And so that's kind of the story here. If you assume that everything interacts with everything, could interact with everything. All treatments could interact with all covariates. That you have a you have a very large number of parameters to estimate if you have a regression model there. And you might use lasso to try to reduce with sparsity to try to identify what are the most active ones. Here, we're trying a little bit different approach. We're assuming we have some knowledge of the structure or some potential interactions, hypothesized interactions, say with Mars3 and aggressive fluids management. Does that help the trial do better? And with Andreas, with Spiros, and with the support of the Amsterdam team, we found that the answer is yes. So our algorithm here is this FEVI. That's the lowest one. Doing also very well is Dan Russo's top two Thompson sampling, a great algorithm. We had some really nice discussions about that. So thank you, Dan, on that. Um, but what this is saying is that you can learn more quickly, get it wrong fewer times. Like if we look at the horizontal line here, we can see for any given target level of expected regret, you actually dramatically cut the number of samples required to get there with this algorithm relative to some of these other algorithms. So this is an alternative to lasso if you have such structural knowledge, as is often the case in biomedical settings. But if you desire, if you get it wrong, maybe bad things happen if you get the assumptions wrong. And so you could add in a lasso heuristic. And so we've shown how to do that, how to handle online rewards, offline rewards, mixed rewards. So there's plenty of fun stuff in there to do around sequential learning when you have a hypothesized structure. So that was kind of fun and a good one to go at. Um, John, do you know if there's any kind of pressing questions there, or should I make maybe a couple of high-level comments about financial incentives in this process?
I don't see any questions in the chat as of now. But
Okay,
for the participants, if you have questions, feel free to put them in the chat. And then I think our plan
yeah,
is to, at the end of the talk, to start answering questions.
okay, cool, yeah. So let me just make a few comments then about money, because that was one of the motivating things here, and then be happy to answer, stick around and answer some questions at the end of that. I'll just give a high level on a couple of points here. Um, so all of the work that I described above, that was all assuming a social welfare perspective. For example, UK funded National Health Service, funding trials, no patent protection to worry about, existing technologies. How much do I spend to learn whether sling is better than surgery? Okay, there's not a new patent protection here. But with drugs, we have it's often with private firms or other technologies of private firms. They have shareholders, they have responsibilities, which are also have finances involved. And so health and finances matter when we have this kind of public-private boundary. So I was super excited that after that initial work had started on these value adaptive trials, value-based, right, rather than cost-focused, and adaptive, being response adaptive. Clinical Trials Insights, which is a trade rag in the pharmaceutical space, contacted me and said, "Hey, let's talk about what is this thing." And so they put an article called "Cash In on Opportunity" on that particular topic. And it's true right now that some of the regulations, I mean, Bayesian trials are okay. You got to demonstrate your power curves today. But even if, and so I haven't talked too much about power curves. A lot of the code will generate some of those power curves for some of the code packages I've described here. But can a firm still use it, even if maybe they feel more comfortable designing a trial with the traditional approach? And the answer is, I think you might be able to, right? Because you could run the trial as usual and then have your stopping boundaries for your own project, and you just stop the trial early if it looks like it's not going to be financially successful, assuming you can gain all the health economic benefits through, say, a Stackelberg relationship, where you're able to take any health gains. You can get 30,000 pounds per quality adjusted life year. So you could approach it like that. I think we wanted to push a little bit further than that. And so we did that with Noah Gans initially, and Özge Yapar, our former PhD student at Wharton, who's now faculty at University of Indiana. Go Red, right? So that's the red team. Can we think about pricing? price negotiations? Can we think about conditional approval? Maybe we're not sure whether the new technology is good enough or not. We don't have enough data. The confidence intervals are big. Should we go collect a bunch more data, validate whether this is cost effective, yes or no, and then set the price? So that's a conditional approval scheme. So I'd like to talk about that first. I don't think I'll have time to talk about portfolios of innovations, but let me just make a few comments there. So conditional approval scheme. So this is something that we've seen emerge in oncology, in particular, for example, the United Kingdom's Cancer Drugs Fund. The goal there is to try to, if there's innovative cancer therapies, can you get them to patients sooner rather than later? If it's starting to look promising, but maybe there's a few more bits of data to go. So this is it's like a third option. After the phase 3 trial, I can approve the drug, I can reject the drug, or I can say conditionally approve. I'll collect a little bit more data, see how the cost effectiveness is with that additional data in hand, and then we'll negotiate on a price. So the process is phase 3 trial, initial negotiation, either immediate approval and I find a price, I reject immediately, or I do that conditional approval scheme, collect a bunch more data. And I'm not going to do this adaptively. We're going to just do one shot. What's the Bayes optimal length of that trial? And then we'll try to do a reappraisal once that data comes in. We'll either reject or approve at a new price. So we need to think here, do we want to run the conditional approval? Do we want to give it to all patients that are in the trial only, but not to anybody else who's not in the trial during that period? Or do we want to give it to other people with that particular condition while the trial is ongoing? So only in research, only with research. The two things I'd like to focus on here are what's the final negotiated price and what would be a reasonable price to pay during this post-marketing trial when we're collecting more evidence. Because I think that's where some new insights come into play when we start to look at this. Because what we found was that there was not a lot of research done in the literature regarding strategic behavior associated with conditional approval schemes. And of the literature that was there, the bulk of that was really assuming a Stackelberg type of a game. Okay, the firm can bring the product to market. A demonstration is given of the health value, and then the firm can capture all of the excess health value to the tune of 30,000 pounds per quality adjusted life year, or whatever your particular jurisdiction would use. And the firm gets all the surplus. And that didn't make sense to us, right? Because, I mean, firms need to sell stuff, and governments like to have stuff for their healthy populations. They also like to have employees working in their country, and so maybe a collaborative agreement can be found. So we looked at a Nash bargaining, cooperative bargaining approach there, and when we did that, well, we found a couple of interesting things. One of them is that we did find a non-surprising result, which is the final price. We found that the cost plus is optimal. So you kind of cover your costs and then you pay extra proportional to the health economic surplus. But what we found for interim price actually doesn't match what we sometimes see in practice. What we sometimes see in practice is websites that will say, for example, the Cancer Drugs Fund website will say, this is kind of targeted towards, we'll set the price during the data collection such that it would just be cost effective at that 30,000 pounds per quality adjusted life year level. And what we find is something very different. That is not enough to align incentives to get drugs to market. It might be better to view the cost of that conditional approval data collection scheme as something for cost sharing between government and company. And with that, it's easier to align incentives in that supply chain. If we put a cap at the willingness to pay threshold, we're not going to restrict too much drugs from coming to market. But if we make a tighter cap, what we found was, in fact, you're going to deter firms from wanting to bring drugs to market if it's a little bit iffy as to whether they might work or not. So that might actually inhibit innovation. But we did find a risk, if we do want to implement such caps, we did find a risk sharing mechanism that will help stimulate through compensation if a drug is eventually approved. So It was kind of fun. It started out as a kind of a sequential learning and stochastic optimization thing and optimal stopping. And when you dive a little bit deeper, you see all these sorts of incentives and game theory things emerge. I don't want to say too much about portfolios of innovations other than to say there's been some great work by Andrew Lowe and colleagues out of MIT around thinking about if you do financial engineering to try to get investments in biotech projects, you're able to bring more projects to market. We've been working with a gentleman in private equity who's interested in really helping to stimulate pediatric oncology investment, and we're on board with him together with my postdoc Zyla Lee, with Shane Henderson, and with Sam Dems, who's working in private equity. We have a paper coming up at WinterSim 2025. So if you want to check that out and see how response adaptiveness might work across biomedical investments in a collection of trials, you can learn about that. But that's kind of the powder in my gun. I've kind of shot the gun there. Um, I'm happy to maybe make just a couple comments about some things I learned along the way. The first one is relate, right? What was really important for me is to take the time to learn the language and context of collaborators. When I started working with Jim Coatman, I didn't know the difference between infected and diseased. And he wasn't always using the words average and expectation, something we might see as very different. So it took us a while we would use the same word, but we would mean different things. And so take the time to get to know your collaborators. Start to ask questions that matter to those collaborators. And then start to question assumptions. Why are those assumptions there? Why is efficiency minimizing the number of patients? Isn't it making it better health decisions? And so what are the logical implications of that? A second observation, reinforce. Don't assume that your base model is going to solve everything. Try to push forward. Have a goal you're passionate about. Really push the boundaries as far as you can. Don't just write one paper, two paper. Just writing one paper to get that paper accepted, it feels wonderful. But find something you love and dedicate some real time and energy to it. And there's value to spending time for a few years on a particular topic. In this case, it's been about 10 for me. And then finally, thinking about rewards and the third re. And I'll give you a bonus 3 ins for this re. This is about, it's kind of business model innovation type of a story. So thank you, Sergey and Karan. Information, right? Information only has value if it can change a decision. So think about expected value of information. That's been a very powerful thing along my journey. I'm thinking about incentives. With the Nash bargaining, financial incentives, those matter. But other incentives matter as well. Some of them are modeled and some of them are not modeled. So it might be you have a fantastic model, but if it doesn't align with some of the organizational issues, like you got to run the trial longer, but my postdoc funding is over. Well, you don't want to you want to take into account that. And finally, enjoy the interface with others. It's fantastic to work with bright people from all over the world, and I'm very thankful for that. I encourage you to, there's a reason why medical journal articles often have lots of authors, and I know many of you also have lots of authors as well. It's fun to learn from each other because it's a bright group and nobody knows everything, and it's fun to share what we each know with each other. So thank you very much. I really appreciate your time.
All right, what a wonderful talk, Steve. I really appreciated that. As of now, I don't see any questions in the chat. Perhaps people are a little bit shy, but actually, one just popped up. So this is from Liang Xu. I'm sorry if I mispronounced your name. And they ask, how will AI change drug discovery?
Oh gosh, that's a great topic, and it's a different topic, although it's clearly going to link in here. Um, I think the Nobel Prize was awarded kind of with this type of theme in mind. I'm not sure that many drugs have already made it to market that were discovered through that mechanism. I mean, this has been around a long time, like combinatorial design of molecules and so forth. It's kind of a rich history with the LLM. I think there's a lot of neat opportunities that are emerging. But it does take, I mean, there is a pipeline, right? It takes several years to get through the phase one, phase two, phase three trials. I did see a BCG report that indicated that the probability of making through phase one trial is higher for AI generated ideas for designs. The success rate for going conditional on getting through phase one through phase two is about the same, whether it's AI or human generated. I think we're still going to have to wait to see what happens there. Um, but you're right, that is an exciting potential opportunity. In the end, we still got to run the trials, though. That phase three trial still has to happen. But maybe we'll be getting better drugs out of that process with that.
Awesome. So I don't see any other questions, but I did have one that I think might be, I think a good question that's a little bit higher level that, especially because we typically have a lot of PhD students or early career scholars
Yeah,
here. But one point you brought up in the very, very beginning of your talk is that whenever you have, you kind of set out to do some research, you like to start with simple models and then kind of build your way up. So I guess
yeah,
like my question is, how do you scope that first simple model and make sure you're answering an important question at the end of the day? And then
yeah,
the follow-up question to that is, do you decide the right level of complexity to add to the next layer? Uh, yeah, so I'll stop my
yeah.
question there and then you.
That's a really beautiful question. And I wish I had a simple model to give you an answer for that. No, yeah, uh, at my time in Ann Arbor, I had a good pleasure of interacting a lot with Steve Pollock, and he was really a strong influence as well. And he was really a big fan for starting simple models. And we co-taught a course that he had designed. He brought me into that around how to develop simple models. And I think having this mindset of a consultant is a good mindset. Like if you only have 1 week to give an answer to somebody, what would you do? And then you make it through that week. Okay, if I only had one more week, what would I do? And I try to do that a little bit. And a little bit as a parallel process, I like to think, well, what is my overarching goal? I want to improve health value for money. Okay, so what would that mean? I know what that means in healthcare delivery. It means something very different in healthcare innovation pipeline. What would that look like? And so I like to learn stuff about bandits and contextual bandits. And I, I don't know. I guess I play a ping pong game back and forth. I don't know if that's helpful or not. Is that how you do it, or are you thinking a little bit differently?
I think that's pretty similar. Like, I think for me, like, because I think maybe this is similar to your perspective, but when you work a lot with like clinicians or practitioners, like, they don't always care as much as you do about the nice math, and what they want to see is like,
Yeah,
does the idea work? Does it, can you
yeah,
give me some proof of concept that this is gonna work? And so like starting simple, even like, you have a week to do it, can you put together a quick simulation or like some visualization that shows the idea that they can hang on to,
yeah,
that gets them hooked on long enough to keep working with you for the rest of the project,
yeah, yeah,
or hopefully many projects down the road. But
yeah.
yeah, I think it is a nice back and forth.
I think listening to their challenges is helpful because it helps you appreciate some of the things that are not in your model, and it's easier to build a trust, I think, with that, and to have an impact if you take the time to listen to their challenges as well. And I think then it's easier for others to listen to what your challenges are. Yeah,
Yes.
yeah,
Yeah, and I think that another nice kind of, I think an important point for me to learn early on too was like the not every paper ends up being like, a super high technical paper. You can answer impactful questions
yeah,
using kind of your basic toolkit as an OR person.
yeah,
And that can pay dividends in the long run, whether it's for establishing collaborations or even just getting the idea
yeah,
out. That's
yeah, yeah. It's something I'm seeing more and more, and I think it's a really good trend. Is a lot of scholars are trying to push towards, okay, here's a theory paper, and maybe I send that to an informs journal, and then here's a practical paper, and it's going to go to a medical journal. And actually, my most cited paper is not in an informs paper, or my second most cited paper. It's in a health economics journal, and there's no new theory. It's just linking how does this model relate to that model, and why should you choose this model versus that model. And that, it's there's not a lot of theoretical development, but there's a conceptual development that helps people in a practical setting. Do I use a system dynamics model, stochastic? Do I use a discrete event simulation? Why does it matter? Um, and that paper has been reasonably well successful because it's addressing somebody's problem. How do I pick? How do I know what model to pick? And can I justify that to a government, the UK government, to the French government? This is a good choice of a model. Right? So that's solving a problem for somebody.
Exactly. All right, so we are a bit over time, and I don't see any additional questions in the chat. So I'd like to thank you once again for giving a wonderful talk. I certainly learned a lot, and I appreciate your commentary at the end. I think kind of this really nice back and forth and tie between scoping the right technical tools to use to answer the high impact questions that are occurring in real life. I think it's just such it's an art certainly,
Yeah,
but I think it's something we can all learn to do.
that's a good one.
Thank you again. Thank you to the attendees for taking the time out on a Friday to join us. Hope to see you all at the next one. So,
Excellent. Thanks, Jen. Thank you, everybody, for sticking around. It's great to have a good group.
have a good weekend, everyone. Bye.
Bye bye.